Hi, I'm Stephanie Jones. I'm a biomedical science major at UWF, and I research molecular markers for Alzheimer's disease. Basically, I take phage libraries, or a collection of viruses with proteins synthesized on them, and I pan them against cerebral spinal fluid of Alzheimer's and non-Alzheimer's disease patients to see if there's any different kind of binding patterns and significant analysis we can take from that. It looks like a bunch of plates with blue dots on them. It's a qualitative measure to see if there's significant binding between a phage and cerebral spinal fluid of control or non-control.
I'm Justin Bobbitt. I'm a sophomore at UWF. I'm studying physics with a minor in chemistry. Dr. Royop is in the copper lab here, and we are doing the synthesis and analysis of copper-1 thiolates. Copper-1 thiolates are precursors or building blocks to nanoparticles. And all of that fancy talk basically means nanoparticles can target specific cells, such as cancer cells, and eliminate them for future biomedical research. I'm working with lab partner Sunny Dawn, and she actually has previous experience in here earlier this year. And she's actually been a mentor to me in helping go through this similar process, but we're expanding upon her previous work.
I'm Lolly Gutierrez, and I'm a biochemistry major, and I have a minor in math. And I'm working on a trifluoromethylator project. Essentially, I'm trying to simplify the process of making this specific trifluoromethylator that can be used in pharmaceuticals and in the making and production and synthesis of pharmaceuticals. I would love to do research in diseases and neurodegenerative diseases particularly. And this project really gave me an opportunity to kind of see how, first of all, it gives me great hands-on experience. But this project particularly, dealing with pharmaceuticals, is something very relevant to my interests.
My name is Jordan Kleinschmidt. I'm a marine biology major here at the University of West Florida, and I am currently studying microplastics in Florida and the effects that they may be having on gastropods. Essentially, we go out into different sample sites and we collect different types of marine snails. Right now we have the crown conch and rock snails that we're looking at. And along with these snails, we take water samples. And when we get back into the lab, we are able to break their tissue down using different nitric acid, and then we dilute it with different water. What we can do with that is filter it, and we are able to see what types of microplastics are actually within these snails' tissues and within the water column. And once we get all of that data together with our numbers, we're able to see what sites may have more microplastics. Maybe these sites are more prone to human involvement and different things like that. With our findings, we should be able to look at where these microplastics are coming from and hopefully encourage the public to really think about where their products are going when they're done with them. Because a lot of the majority of these microplastics are coming from breakdown of beauty products, leftover beach toys, and different things like that from restaurants and such. And if we can see where the sources are, I really do think that this project could inspire people to maybe cut down on their plastic usage.